I'm Scarlett Norris, and this summer I did a dendrochronology intensive summer course in Tucson, Arizona with the University of Arizona. I would like to thank the archaeology department for funding this project, and then another project I did this summer with Archaeology Southwest that won't be in this presentation, and then also Dr. Campbell for allowing me to do research, dendrochronology research for him, and giving me this opportunity and telling me about it. Okay. So we were doing work in the Verde Valley in Arizona. This is just like various different sites. We started here. There's another map. But basically we started out in the lab, just getting an overview of what we were going to do. Then we collected samples, and then we came back and it was preparation, sample preparation, and then sample analysis. This is just another picture of the area where we were. We collected dendroecological samples from Mingus Mountain on the map, and then we also collected dendroarchaeological samples from Sycamore Canyon. Both can be used for archaeology. The dendroecological samples are taken from live trees. It's a tree core, and then we can compare like what we see in the tree to human activity. But then the dendroarchaeological samples are from like archaeological sites, and we try to make inferences on what was happening when sites were built, before they were built, that kind of thing. So like I said, this is Mingus Mountain where we took the dendroecological samples. We also visited various sites around just to see the sites in Arizona. These are two sites on Mingus Mountain. This is kind of the overview of everywhere we started in Camp Verde, and then there's also Mingus Mountain is on there, and then Sycamore Canyon up there, and then the rest are mostly just sites that we visited. The kind of like key point of interest that we were looking at on Mingus Mountain was a power line that was put in. So that was the human activity that we were comparing to the live tree cores that we took, and to see if we could find any evidence of the disturbance in the tree cores that we took from the trees. The species of trees we were doing for the ecological samples were ponderosa pine. So we found basically like through historical records that the power line was in the 1930s, but then also some of it was like 1990s, but we overall I think settled on around 1990s, 1980s. So on the site in Mingus Mountain where we took the live tree cores, there's the first line which we took that's from the trees that's right next to the power line, and then we went 50 meters back to take like a base sample, like so we would compare the two from sites like A and B, but it was all within 50 meters of each other. This is how we take tree cores. We just kind of, you put it in the tree and you twist it. For the archaeological samples, it's different because the wood, it's dead wood, and so it's harder to take a sample, so you can't just like twist it in there, but we'll get to that. This is, yeah, we were comparing this sites A and B. We also took stump samples to see if we could date those and if they were cut because of the power line. This is like this is what the archaeological samples look like. We were at a cliff dwelling in Sycamore Canyon that I can't reveal the location of because it's just not necessary information. It's not, the archaeological samples are bigger, and these were also cottonwood and not ponderosa pine. Cottonwood is harder to date, and then we also just don't have as long of a series of our core. It's also not from like, we might not have bark on one end or the pith of the center of the tree on the other, so it's harder to date. And overall, we ended up having to, we didn't date these cores in analysis. We had to send them out for carbon-14 testing. It's called, like, it's a new carbon-14, it's called fill matching. But we would take them from like lintels in the structures or different support beams. So basically, just how you prepare and prepare the live samples and the archaeological samples. We take them, then we sand them. We have to mount them as well. Mounting is just like we use Elmer's glue and string, and then we dry them, and then we sand them, so we can get a clear look at the cross sections of the trees. There's like standardized markings that we do when we're doing analysis. to count the rings, and sometimes there's things like false rings or missing rings, and we have to account for those. How we compare them, there's chronologies, existing chronologies that are in the International Tree Ring Data Bank. So we can pull from different data that is out there about the trees and their rings and dates for them. So we did that. This is more of that, where we put in the samples that we had and other samples that are out there. And then we use Cofetcha. It's a program that tells us like series correlations and that sort of thing. And then this is like a general correlation, but there's letters next to it, and those basically indicate, there's years and the letters indicate the width of the ring. And then for each year, that like the highlighted ones are ones that stand out as either really like skinny or really big. Really big tree rings is like the growth rate, and that's usually in like a wet year or like standard year. If it's really big, there's we mark that down, and if it's really skinny, it's usually like dry, there wasn't rain, that sort of thing. Then we made our own correlation based on the samples that we took from Mingus Mountain, and these were the patterns that we saw for them. So specifically, when we look at a tree core, we can, if we're seeing this, we can make an inference like, okay, 1904 is a very narrow ring, and we can look at and say, okay, maybe this is that, and then we compare to the other samples that we have in our overall chronology to check that. 1929 and is also narrow, 1920 is wide, so these are, you can see like the different marking patterns, like for decades and stuff on there. Just more patterns. So this is one way that people analyze tree rings, is called skeleton plotting, where basically you mark lines for the higher the line is, the skinnier the width of the tree ring is, and then we can look at those compared against larger chronologies and match like an unknown tree ring to a date. There's a practice that's online, like if anyone's interested, you can do it online for practice. Yeah, this is just marking the grids, and then this is like a chronology, a known chronology that we have that we can match up our single core with. In lab, we practiced on a Zuni collection where it was just skeleton plotting practice. You can see it's a good match. Oftentimes it's criticized for being pretty subjective because you have all different kinds of people just going and making marks based on whether they think a ring is skinny or not, but it actually does match up. These were 3 different students from the program, and you can see that you can match them pretty well based on they correlate. This is more on sample preparation. We go and we take the live tree cores, we put them in straws, we label them. That's like in the field, and this is showing the process of mounting them. It's really important to align them correctly. And then we would go in the wood shop and we would sand them. Yeah, and then these are just some pictures from, this is the lab that we would do the analysis in, and these are some photos from the field. Yeah, then I can talk about results really quickly, but basically we have different graphs. This shows a 20-year moving correlation across the trees from Mingus Mountain. These are all results from Mingus Mountain because we didn't get the carbon-14 dates back from archaeological samples. But this one up here is a comparison of the sites A and B, and we can see in 2000 for the edge trees, which were along the power line, there is a disturbance. It's just intervariability within the edge trees and the interior trees. And so this is like a typical negative exponential decline in the correlation that we would see. So like all the trees are similar to each other on the interior, and then the edge trees were, there was some sort of disturbance, which was, we're guessing, maybe the power line. And then, yeah, this is just more showing averages. This is also showing the intercorrelation between the trees. That is the like standard average line, and you can see a disturbance too here in like the 2000s, but it's going back down to the normal variability. Okay, so that's it.
Questions for Scarlett?
I'm gonna ask a little more about your results, if I'm allowed. What is kind of the working hypothesis on why those tree rings would have gotten wider in the edge trees?
So it's not necessarily that this isn't about like the width of the, we can tell the differences based on the width of the tree rings, but it's not that they were getting wider. They were likely skinnier for that moment because the cells that are in is what we look at when we see when we look at them up close. We can see the cells and each ring. There's late wood and early wood. Sorry, I don't even have a good example picture. Like these, this is the late wood and this is the early wood. So maybe for here, we would think that there was some sort of disturbance or it was a really dry year or something because the tree wasn't healthy enough to produce like all of the cells that it would have produced in like a normal, relatively normal year. So we can tell based on that, and then it would just indicate a disturbance because one, we know that it was there, like the power line was put there at that certain time, and if those trees, if they're returning back to their normal correlation, then we can assume that maybe that was something that affected their growth. I don't know if that probably wasn't like a good explanation.
Do you ever need to like differentiate between shorter growth periods because of like disease or parasites versus climate?
You can usually tell. I didn't include it in here, but if there's parasites usually only go so far into the tree. It's like past the so there's bark and then there's cambium. The parasites usually go in there and they'll die in there, and you can tell based on like how it looks. If there's scarring, you can tell if it was like a bug or climate or something like that.
Thank you, Scarlett.
Thank you.